everyone, and thank you for joining this event, which is part of the 2024 edition of the European Green Week, the annual conference on environmental policy organized by the European Commission. This year's edition focuses on the critical challenges facing our water resources and the urgent need for a European water resilience. I'm Laura from Mhamad, one of the Imptox project partners, and today I will lead you through a series of presentations by our esteemed researchers. Our partner event today is dedicated to the presence of micro and nanoplastics in our waterways that pose a significant danger to aquatic ecosystems and potentially even to human health. Our researchers will present their latest findings on micro and nanoplastics, and they will cover a variety of topics such as co-exposure to cyanotoxins, microplastics as event for microorganisms, and the transport of pathogenic bacterial and metal ions by microplastics. Before we get started, I would like to go over a few things so you know how to participate in today's event. Regarding the setup, all participants will be automatically muted and the event is recorded. At the end of each session, participants can interact with us by submitting questions through Slido. You can scan the QR code behind me on the screen, or to access Slido app and insert the access code that you find in the chat. If you encounter any difficulties, please write in the chat and we'll be more than happy to help. Now, I would like to present our first speaker of today, Professor Tanja Cirkovic. She is the Imptox project coordinator and the full professor of biochemistry at the Faculty of Chemistry of Belgrade University. She is also the coordinator of the university's Center of Research Excellence for Molecular Food Sciences and the corresponding member of the Serbian Academy of Sciences and Arts. She serves as a member of various scientific organizations and editorial board member of a variety of international journals. Professor Tanja's presentation today is Imptox project research on micro and nanoplastics in water environment. Tanja, the floor is yours.
Thank you, Laura, for a very nice introduction, and I thank all the participants of today's workshop. I'm going to give an overview of our research on microplastics in water environment, in particular in relation to the microplastics we have identified in different seafoods. And I hope that other partners of Imptox will later on give more information on different aspects of this emerging environmental pollutant that we are analyzing and working on during this project. We all know that microplastics is abundant environmental contaminant, and that only in the oceans there could be more than 5 trillion microplastics circulating. The forecasts, at least for the Eastern Tropical Pacific, show that the numbers of these particles will exponentially increase in the coming decades. And if we are at a level of around 100 microplastics per cubic meter, the numbers will double in the coming few decades. And of course, one of the main reasons for this is that plastic litter is everywhere, and also plastic is a material that is durable. It's made to remain unchanged in the environment. So saying that public interest is high in this topic is an underestimation, definitely. All recent research discoveries, all important research findings of microplastics in different environmental compartments and also in biological fluids and tissues of animals and also humans is immediately reported to the public. So everyone already knows that microplastics particles are found near top of the Mount Everest, in Siberian rains, also in unborn babies, in placenta, and they are most of the time informed through news and magazines directly from the headlines. So where does it come from? Microplastics can be produced as a primary microplastic. So if it is already produced as this tiny particle that we define as a plastic material that might contain different additives, and it has size of up to 5 millimeters, then we can say it is already produced as a microplastics. It comes in different forms, shapes. Also, it can have different chemical composition depending on the polymer type, plastic polymer type it is made from. And of course, it can contain a variety of additives such as stabilizers, pigments, and plasticizers. Means that this is a highly heterogeneous group of particles, and its chemical composition is very complex. It also means that analyzing that kind of a material is chemically extremely challenging, and I'll briefly also say a few words about this. Microplastics in the environment can also be a product of plastic degradation. It means that any plastic litter, being it some larger plastic products such as PET bottles, or maybe clothing that is made of synthetic materials, even the small plastic pellets that are used in industry, or maybe cosmetics, microbeads we find in cosmetics, is subject to environmental degradation and the influence of UV radiation, mechanical action of waves, also the microorganisms we find in the environment. And already existing plastic and microplastic can further degrade and also change. This process of aging of the plastics and microplastics we also call weathering, and this fragmentation of the larger plastics also leads to the microplastic we find in the environment. It means that the material we encounter in the environment and in the biological fluids, also in foods, has a kind of irregular surface most of the time, and it is also very challenging to be analyzed by analytically available equipment. Well, we can see we have seen expansion of research on microplastics since 2009, since we probably first time mentioned that there is a release of these tiny particles into the environment. There is an exponential growth of research on the topic. Of course, with the findings that the microplastics can also enter into the food chain and also get into the biological fluids, we all really want to know, and the researchers are putting a lot of focus on probably the main question related to this emerging contaminant. So is there a risk to environment and human health because of the omnipresence of these tiny particles? So in order to estimate risks to the environment or to the human beings and also animals, of course, we need to have information about the extent of exposure to the microplastics, as well as the hazard associated with this material. And the existing information on exposure levels for now exist or are available related to the cosmetics, that is that we are exposed to via skin, to the plastics manufacture that we can inhale through the air sprays or air, maybe some additives that we ingest via ingestion. But most of the exposure information on the degradation products of microplastics, the aged microplastics, is still research in progress, and we are experiencing a lot of difficulty in collecting all the relevant exposure information. We are also working on the hazard aspects of the plastics, and the information will eventually lead to the proper risk assessment that will help us really understand and regulate these contaminants. Within the Imptox project, we are trying, of course, to improve our exposure assessment and to understand the levels of the exposure to micro nanoplastics, particularly in food, but also in water and air. We are also trying to evaluate the role of the micro nanoplastics as vectors for contaminants, and some of these contaminants we find in the water environments, pathogenic organisms, viruses, bacteria, also toxic metals, and we will hear later more about these aspects of Imptox research. When it comes to the ingestion exposure, there are four main routes of the exposure. The microplastics can get into our food, and we can ingest it through the different food additives such as salt and sugar, drinking water and beverages. Also, the microplastics incorporated into the food chain, and the migration from plastic packaging during the food processing. And there are different studies reporting the levels or average levels of the microplastics found in these sources. So when it comes to the human exposure, already based on the insufficient, or let's say incomplete data, the scientists started modeling the human exposure. So a few years ago, we have seen the models that predict that we ingest around a credit card of plastic per week in the form of microplastics, meaning around 5 grams. So these models and predictions have been made on the, as I said, not so complete information, and also the modeling applied was questioned by other researchers. And according to different models, we do not ingest 1 credit card of plastic per week, but maybe more than 1,000 times less microplastics. So these are the ranges. You can see 10 to the third are the differences in the models used to predict the human exposure. It's a huge range. When it comes to the topic that we are researching, the seafood, or let's say food as a source of microplastics, what we have seen, particularly when it comes to the seafood and shellfish, that most of the studies reporting and looking at the looking at the quantities of microplastics in seafood are based on the material collected from the environment, from the field studies. Not so many studies are based on the material that we can get from the market. And just to kind of introduce this topic, well, the microplastics analysis in food is quite complex because there are few steps that have to be done before completing the analysis. And the first one is the matrix digestion, because microplastics in foods is present in the biological material that should be digested, so it's very complex. Then the extraction process, the counting of the particles, their morphology characterization, and the chemicals characterization. So it has to be a full characterization, and one of the most common techniques applied for this kind of research is infrared spectroscopy in combination with microscopy, so the so-called micro-FTIR or micro-Raman. And then we have this full picture for the presence and the numbers, and also the characterization of these particles. I just have to say that there is a size limit of the most commonly used method, micro-FTIR. It's a 20 micrometers. The materials such as nanoplastics or very small microplastics cannot be, it's practically invisible in this analysis. There is a lot of analytical challenges, not only related to the material itself, which is complex, should be degraded before the microplastics is extracted. We also are facing analytical challenges when it comes to the method we are using for the quantification and chemical characterization of the instrument. And I can only demonstrate this in a report done in 2021 from GRC, And Beaumont, an interlaboratory comparison study on PET in vulture water, is relatively simple matrix, and PET is only one type of plastic material that is present in PET bottles. So this interlaboratory comparison study has shown that around 100 labs that participated in this testing reported values when it comes to the quantification of the particles in liter that goes from 0 up to few thousand particles per liter. So the range of the values that they have obtained is enormous, and it's something we do not frequently see in the interlaboratory comparison studies. Well, in our study, we wanted to minimize this kind of methodological issues, because we wanted to analyze point of sale samples such as mussels, clams, and shrimps in a single laboratory. So we collected the material from different markets, from Belgium, Croatia, South Korea, from our partners there, and also some of the samples were from our market in Serbia. We have worked on improving the microplastics extraction protocol in a way that we finally end up having a very neat surface for the chemical analysis that will allow us to actually extract and quantify all the microplastic material in a single sample, and the single sample is a single organism. So we could report our values as a microplastics number per individual. Our idea was to compare different markets. We know already that in the Mediterranean area, there is a lot of microplastics in the sea. We also have heard that maybe the South Korean seas are also heavily polluted. So we wanted to check, is it possible to see difference in the number of these particles in the point of sale samples, in the market samples that we eventually eat and use. And we, as I said, employed micro FTIR to quantify and fully characterize these microplastics. So our numbers and figures we have seen, in fact, has shown that not in all of the material we analyzed, we detected microplastics. Some of the samples we literally could not extract and quantify a single microplastic. So around 30% of all samples was microplastics.
So maybe we can postpone the question session referring it to Tanja presentation and go ahead with the next presenter, Professor Andreja Rajkovic. Andreja is an Imptox investigator and full-time research professor at Ghent University. He is a president at the International Committee of Food Microbiology and Hygiene, a member of a scientific committee of Federal Agency for the Safety of the Food Chain in Belgium, and he is an expert in the Superior Health Council in Belgium. He is an editorial board member of a variety of international journals and a founding editor-in-chief of the International Journal for Food Safety and Risk. So I have the pleasure now to give the floor to Andreja for his presentation. Thank you. The presentation of Professor Andreja of today is entitled Risks of Co-Exposure in Cyanotoxin and Micro-Nanoplastics for Irrigation Water.
Thank you, Laura, so much for the introduction. Thank also for all the participants being here and our previous talk of our coordinator, Tanja. I will go today a little bit into something which is really close to my heart, something that actually we are investigating very heavily within the work package 5. Actually, all the presentations of today within Imptox are situated in the work package 5, which talks about the risks and exposures to micro and nanoplastics in relation to other co-contaminants. And the co-contaminants I have selected today to talk about, because it's very much related to water as well as to climate change, for example. So the resilience of our water systems in general is the risks of exposure to cyanotoxins and the micro and nanoplastics from irrigation water. Now, of course, you may wonder, why am I talking about this particular combination? There is a good reason for that. Basically, we are all aware that there is a very certain and very growing evidence of the presence of nanoplastics as well as microplastics in water. We have been seeing some data now shown by Tanja. You've been all seeing the images on different major media as well as on social media showing and talking about the plastic pollution of the water systems, but also of the soil and the agri-food chain in general. Now, when we talk about water, water is far more than what we actually drink. Just an example relevant to this presentation is water used for the production in the agricultural sector as well as in the food industry. So we know that there is microplastic there, we know that there is a nanoplastic there, and what we wanted to know, is this something that can be transferred from the water into the plants? We talk about plants as a future food. We are talking about the protein shift. We are talking about the dietary change. So we need to take a look also into potential risks related to that if we want to give fair and informed advice to the consumers and to the policymakers. So within Imptox, we looked also into the effects of contaminated by micro and nanoplastics irrigation water and their uptake into the plants that are irrigated with this water. Now, we know that the climate is changing. We know that there is this global warming issue, and we know that there is already an established evidence of potential presence of cyanotoxins in water bodies used in the agri-food sector. Now, while you can imagine nanoplastics, it's something which is very much somehow visualized in our minds, though we cannot really see it, I want to draw your attention what I mean by cyanotoxins. There is no way anybody in the world has ever not seen this greenish layer with bluish tint on the water. And this water is actually any water. So the lakes where we swim, the lakes and the rivers along which we are jogging, the water we use for recreation, the water we use for irrigation. So just in the city of Ghent, where I'm, every year almost, we see on the major recreational domain these greenish layers. And those are the cyanobacteria, or otherwise known as blue-green algae. And they can produce toxins, toxins that we call cyanotoxins. The cyanotoxins, if they are in the water, they are going to enter the food chain, either of the animals and plants situated in the water or in the land materials, like agricultural crops. When we irrigate them with the water that has these cyanotoxins in it. So this is how we actually see the whole transfer from the water contamination into the actual agricultural and food chain. It is one of those examples. Now, with the growing human activity, with the changes in the climate and the global warming, with the irregular raindrops, we actually see this problem becoming more and more present and taking longer times of the year than it was previously thought. So there is no surprise, basically, that we have seen, and other ones as well, like I'm showing here on the slide, that fresh produce and the soil can accumulate these cyanotoxins. Now we wanted to know, how much does this really happen? What's the dynamics of it? What is the mechanism of it? So we first look with the cyanotoxin, the one that is most important, the one which is called microcystin LR, which is also the only one which is really legally regulated because of its toxicity and because of its prevalence. So what we did a couple of different experiments, and I'm going to guide you a little bit through so that you can get a picture of what we've been doing. So we've been growing basil in the hydroculture as well in the soil. Here I'm showing the images of the hydroculture basically, and we kind of gave the basil its nutrients and water through the water that was not contaminated and the water which was contaminated with microcystin LR. Remember, the most important cyanotoxin. We did different concentrations of this, even up to 100 micrograms per liter, which is a kind of a legal limit for certain things. And we actually came out to conclusions. So what we first thought was, will there be anything on the basil plant that will alarm us, that will indicate potential presence of these toxins? So if you take a look at this slide and compare the blue dots and the red dots, where the blue dots show the basil plant before the treatment and the red dots show the same plants after the treatment for different concentrations, you'll see basically no difference. So that means that the basil plant is not going to alarm us. It is not going to tell us that there is something on them or in them. So we will be consuming those leaves without any warning. So, all right, that was good to know, a bit scary, but we had to figure out, are there now really a lot of those cyanotoxins in the plant itself? So we started to take a look. We took a look actually the total microcystin concentrations when we actually gave the water contaminated with cyanobacteria containing all sorts of cyanotoxins inside to the plants, and also really well-known concentration of our really focused molecule, which was this microcystin. What we saw that depending on the concentration we used, we show certain uptake. The lower the concentration we had in the irrigation water, the smaller the uptake. The higher the concentration, higher the uptake. So that already gave us an indication of potential mitigation strategies, which was try to not have the cyanotoxins inside of the water you use for the irrigation, if you can. Now, another important aspect was knowing the plant was, will this toxin be in the leaves, which we normally consume, or will it be in the roots? And the majority was, luckily for the food consumer, in the roots, which are normally not consumed. Now, in the circular economy, you also don't throw away the roots. So the cyanotoxins will end up somewhere in the food chain in some organisms. But for the food consumption, the direct one, the leaves are the most important. So although there was a smaller concentration of leaves than in the roots, there was still some and significant amount of cyanotoxins. So that's already good to remember. We tried the same thing, but on a large scale, semi-commercial scale, major glass houses to do this in lettuce, carrots, and strawberries. And we gave them also the nutrient solution spiked with a known concentration of microcystin. And while we found no uptake of this molecule in the edible parts of lettuce and carrots, we did in some non-edible parts, though, we did find quite some in strawberries. You know, for many of us, one of the favorites, red berry fruits. We have found a lot of it actually in greens and the roots, but that we don't eat. Again, circular economy issue, this does end up somewhere in our food chain. But in the strawberry, we did find as well. The amounts were not always the same. They depended on the type of the agricultural practices we applied. In this case, irrigation method. Whether we use the drip irrigation or the overhead spray irrigation played an important role. That shows another potential mitigation strategy. By adjusting your agricultural practices, you can avoid or limit at least risks to the exposure and the uptake of these toxins in the berry fruits at least. Now, these amounts, they mean something, right? We talk about here very small amounts, basically nanograms. And I'll tell you a little bit about it in the next slides. Now, you know, risk and hazard are not the same. Risk requires that the hazard is present in the food or whatever we are consuming or breathing in or drinking, but we also need to take into account the consumption part. How much of these particular red fruits are we going to eat? So just a back of napkin calculation, do not take it really as an important number, but shows you that if we eat so many strawberries and related red berry fruits, we are going to reach only about 6% of our tolerable daily intake of these toxins. For the children is a little bit more because of the smaller body weight and maybe the amounts they consume, but this is a fraction actually of what we are allowed to take, so to say. Nevertheless, a little bit here and a little bit there, you may reach dangerous amounts, keeping in mind. So what did we see with when we irrigated basil with a nanoplastic containing water? So when you compare this left and right side, you see the left plant with no nanoplastics, the right-hand side plant with the nanoplastics. No really big difference. I mean, you see some difference, but I mean, would not stop you from eating. That was our impression, and maybe these images don't show this very well, but this was the case. Now, our data that just came in last week, for which I thank our colleagues in Belgrade, we show that, give or take, about 60% of the supplied nanoplastic material ended up in roots, very small fraction ended up in stems and leaves. So that means about 5 micrograms of this nanoplastic that we supplied ended up in the roots in the totality, and very, very, very small amounts in stems and leaves. But these numbers, though small, need to be checked. Are they toxic? So we looked at the toxicity of these. We did a classical cell-based endpoint toxicity. We saw really very few toxicity there, so you could say no risks. But we wanted to look deeper into that, and we went to look into the mitochondria, you know, the power plant of our cells. That gives us the energy. If that doesn't work, our cells won't work. They may actually become very dangerous to our own health. So we did this with a very specific techniques that we have developed in our lab, and we found something very, very interesting. So bear with me for about a minute to guide you through these slides. On the left-hand side figure, you see our intestinal cells used as a model for our intestines. What we show is when we expose them to nanoplastics and cyanotoxins, or apart from each other, we see that these cells under certain conditions and certain concentrations change their profile. What we show is that only at a very high concentrations, we talk about 10 micrograms per milliliter of nanoplastics, so much more than what we had, not much more than what we had on the previous slide in Basel. Regardless of the amount of cyanotoxins, we see that our cells are changing. So the toxicity and the metabolic shift to not a very good glycolytic profile, which is very inefficient, looks a little bit like a cancer type. We show the effects of the nanoplastics on the cells. At the liver cells, this is the right-hand side graph, we saw the similar thing, only at a 10 micrograms per milliliter of nanoplastics. Again, pretty much regardless of the microcystins presence, we saw the shift from a very active cell to a very dormant cell. Again, not very good, not for a liver which needs to break down all sorts of toxins in our body. We look deeper into that. Why is this happening to our cells? We saw that actually in the CACO2 cells, basically the most important parameter, which is a basal respiration, so the way our cell on the basic conditions produce the energy, is not really influenced by the microcystins, but it is influenced by the concentration of nanoplastics. This is this left-hand side here. So you see, here we have situation that we see the drop in the basal concentration based on the concentration of microcystins at 1 microgram per milliliter of nanoplastics. But when we increase the concentration of nanoplastics to just 5 micrograms, you see this major drop. Look how less well those cells, those mitochondria perform. And when you go to 10 micrograms per milliliter, which is still very few, basically, you see even a bigger drop. When we take a look at the liver cells, we see, however, in this very specific way of studying, that even microcystins plays an important role. So you see this best here if you compare 3 different concentrations of microcystins, right, with the same 10 microgram per milliliter of nanoplastics. So that's really important to remember. We see that although our cells are not dead, they are actually getting changed. So as the final slide, let me give you a take-home message. Water is much more than meets the eye. It's an ecosystem with multiple hazards, but the hazard is not the same as risk. Risk is a statistical category that combines different aspects. There is an uptake in the plant, so we should not be ignorant of that. The concentration will be an important factor. The edible parts will be important, and how much we consume will be another important factor. Unfortunately, nanoplastics don't come alone. They are together with some other contaminants, and this increases the toxicity. How toxic will it be? Will be dependent on the organ. Intestines behave differently than the liver. And finally, to maintain the water resilience means to maintain the eco-safety, food safety, and public health, as well as an economy. So the fastest and the shortest things we can do is to reduce the exposure. It's a different question, how can we do that? I thank you for your time, and thank also our European Commission for funding this Imptox project, but also the Belgian Federal Service for Public Health, who funded part of these experiments, especially with carrots, strawberries, and lettuce. Thank you.
Thank you, Andrea, for your insightful presentation. I can start reading the questions. This is probably a question for Andrea. Given the co-exposure to cyanotoxins and micro nanoplastics, what could be possible mitigation strategies to reduce health risks, and particularly in agricultural contexts? And you have another one. Maybe I give you both of them and you can answer. What are the long-term ecological implications of co-exposure to these pollutants in irrigation water, particularly regarding soil health and crop safety? Thank you.
Well, I think, I mean, we got off with a quite difficult questions, but really to the
We
point.
have a very few minutes, Andrea, so.
On the first question, what I can actually tell is that the mitigation strategy will have to be multifaceted. On one side, it will probably need to come from the increasing the awareness of both of those who are on the production side of plastics to figure out and invent better ways to use the degradable or faster degradable plastics, as well as those people who are finding the way to degrade them, for example, through the use of microorganisms. Second thing is that we would need to be very careful the way we go about our water bodies. I mean, not only we need to reduce the contamination, but also the water needs to be used in a particular way when you know that the contamination potentially is present. Another thing is that we also need to investigate further how much those mitigation strategies that we call food processing have an impact on the final concentration and the type of the micro nanoplastics found in the final food product. That means that actually what we have on our table is not the same what we harvest on the field. And there will be most likely a very big reduction factor from what we have in the contamination terms on the field and what we have on our fork. So I would say to this in the sake of time, but I mean, there are other ideas as well. On the second question, what is the long-term effect on the ecosystem health? Well, to be honest, that's something much more difficult to say because we don't really have the right controls. So we don't have the situation which is absolutely pure of microplastics and nanoplastics and say, well, look, if you don't have them, this is what is going to happen. If you do have them, this is going to happen. But we can, of course, go way back and compare with the situation the way it was before. But here we have a problem. The crops yields 20 or 30 or 40 years ago were far lower than what we have here. So the level of yield, I don't think we have a problem. What we might have a problem with, and this is what I think about the total quality of our food production, is that with the climate change, the global warming, the need for irrigation is higher. And if you have to irrigate, you're doing this to save your yield, to bring the product on the market. If you are a farmer and if you are a consumer, you would advise for that because you want food. The problem is, what do you do if you have to irrigate, but the only water available is the contaminated one? If you don't irrigate, you lose the crops. If you irrigate, you endanger the public health. And this is where the science and the policy makers, as well as the food business operators, join their hands and they try to figure out, do we need to change the type of the crops? Do we need to change the locations where we grow them? Do we need to think on having a preventive ways that will tell us in advance when those blooms, for example, of algae are going to happen? And how can we actually filter out the majority of those nanoplastics, for example?
Thank you, Andrea.
This answers part of the question.
Thank you very much. We have another very interesting questions. Someone pick what you just said. So can you conclude what effect do nanoplastics have on the performance of the mitochondria? And what does it mean for human health?
Nanoplastics at even very low concentrations, while they don't kill the cell, they impact the mitochondria, making them shift from the oxidative phosphorylation, which is a very good, very efficient, most healthy way to produce energy, to something which is called glycolysis, something which is far less efficient, makes your mitochondria maybe flexible to do that, but it's not a healthy situation for the cell. That's just saying that the healthy cell normally uses the oxidative phosphorylation. The cancer cells typically uses the glycolysis, creating acid environment around it. So the nanoplastics does have an impact on the mitochondria, keeping in mind that I haven't told you anything yet about the possible effects of other nanoparticles. Nanoplastics is not the only nano thing we deal with. So there are still things that I could tell you on it, but let's put it here to an end.
I would like to present our next speaker, Dr. Elsa Gadoin. She is a postdoctoral research at Ghent University. She is a marine microbiologist with a focus on viruses and bacteria, with a strong background in molecular biology and oceanography. Elsa is studying now the formation of biofilms on microplastics particles and by natural planktonic communities. Her research aims to better understand the diversity of species and genes attached to microplastics in aquatic ecosystems. Today, Elsa will present the microplastic in aquatic environments, heaven for microorganisms. Elsa, the floor is yours.
Thank you, Laura, for this nice introduction, and thank you for everyone in the audience for joining this session today. So we are going to speak about microplastic in the environment and the consequence they might have on those ecosystems. So as we previously said, plastics and microplastics are ubiquitous in our environment these days, because we are producing more and more plastic this year, in the years, every year, and we are recycling them not enough, which end up to the accumulation in the environment, and mainly in aquatic environment through riverine transportations and domestical input. So I would like to see with you why we can say that microplastic are a threat to the aquatic ecosystem, and there is several reasons for that. The first one is the direct toxicity of the plastic themselves. As you know, it is made of polymers and different kind of additives, and many studies demonstrated that those chemicals are toxic to human, to animals, and recently, there are more studies demonstrating that they are also toxic to the very basis of the food chain in aquatic ecosystem. As you can see on this publication, it can affect the photosynthetic activity of phytoplankton and algae, but also affect the growth and development of very small organisms, which are the marine copepods, and which are the first consumer of phytoplankton in the food chain in aquatic environment. So this is the first reason why plastics are a threat for the environment. The second is maybe the most known, which is the risk linked to the ingestion and accumulation of those components in the food chain, in the fish that we eat and the seafood. I choose to show you this picture of copepods ingesting microplastics, because it is not only the seals and the sea turtles that are eating the plastic. The very basis of the food chain are eating also this plastic, all the way up to human consumers. The third effect is more indirect. Microplastic, as well as larger plastic fragments, have the ability to absorb the different pollutants that might be present in aquatic environments. So we are speaking here about heavy metals, hydrocarbons, antibiotics, and pesticides. And because microplastic are very light and travel easily on long distance via currents and waves and winds, they can participate to the spreading of those pollutants in the environment, with some consequences as well on the fauna living in those environments. But also the microplastics are able to be colonized by microorganisms, and this is what we are going to speak today. When they enter the environment, microplastics are quickly colonized first by bacteria and then by other kind of microorganisms, forming a dense structure around the particles, and now it is commonly referred as the plastisphere. So it is all the kind of microorganisms that live attached to the plastic and microplastic. So why is it a threat to the environment? Well, first, because this colonization of the particles might have an effect on their transportation. So they might change their buoyancy and lead them to sink while those plastics were potentially supposed to float. And it also favors the transport of microorganisms attached to the plastic from one ecosystem to the other. Among those microorganisms, some studies have identified some potential pathogens, such as Vibrio, Listeria, or Salmonella. Maybe those names ring a bell to you. So here again, those potential pathogens might be transported from one ecosystem to the other and might threaten the biodiversity. Also, recent study demonstrated that in the biofilm attached to microplastic, there is a high rate of what we call horizontal gene transfer, which you have a picture here. So it's basically a process which is very common in bacterial community, where the bacteria will exchange some genes because they are close to one another, and they exchange genes which are useful for them for their metabolism, their survival. But it is a problem when they exchange what we call antimicrobial resistant genes. I think you might have heard about it. Those genes help the bacteria to resist to the antibiotic that we use in medicine, in agriculture, aquaculture, etc. So this might be a problem because of the transportation. If those antimicrobial genes are exchanged in between the bacteria inside the microplast, inside the biofilm, they might be transported and spread into the environment from ecosystems which are close to the coast to more pristine environments. So this is why in the Imptox project, we aim to characterize the microbial communities forming at the surface of different types of microplastic. And we had several questions. The first one was to have a look at the composition of the biofilm. We wanted to answer the question, who is there, but not looking only at the bacteria, because many studies started to observe the diversity of the bacteria attached to the microplastic, but also including the eukaryotes and the virus to have a more global picture, like to really see who is there. And we were also interested in the genes they do carry to have a more global picture of the diversity of the resistance genes that are among the biofilm, but also of the virulence genes. And we were interested to see how those community composition vary according to different parameters. So how they vary according to the site they are found in, and also the duration of their incubation, because we know that microplastic can stay for a very long time in aquatic ecosystem. So for that, very briefly, we had developed an experimental design here in the North Sea coast in Belgium, in Austin, where we used a kind of tea bags to incubate the microplastic and collect them later. And we incubate them in different locations around Austin. So we had freshwater ecosystem and seawater ecosystem, and we let them for 1 month incubate for 1 month incubation and collect the plastics to identify the different taxa that we found attached on it. So the first thing that we saw is an incredible accumulation of microplastic around the particles. As you can see on the pictures on the right, you have the particles, and with the red arrows, I designated the dense structure of microorganisms that are attached to those. It's clearly shown on the graph on the left. You can see that the blue and reddish bar plot are showing the bacterial abundance that we had in the biofilm, which is way more important than the bacterial abundance we found in the seawater, which are the really little blue bar plot that you can see on the bottom. So we saw that the plastisphere and the biofilm are actually this very dense structure around the plastics, with also some phytoplanktonic cells that you can see on the picture on the bottom. Those kind of change are phytoplanktonic cells. Then we were interested to see how the composition of this variability of this biofilm varies. You have on the first graph on the top the bacterial compositions in the biofilm at the different sites. So you can clearly see that we have different colors, different proportions in those colors, and they represent different type of bacterial class. So we see that the composition of the biofilm is very different from the composition of the bacterial communities in the water, but it is also very different in between the sites. So it is showing that at each site, we have a specific bacterial community attached to the plastic, which pose a risk and the question of what happens if those plastic migrate to another ecosystem. Do they bring the bacteria with them, and can those bacteria implement in a new ecosystem, and what are the consequences of such transportation? When we look also at the diversity of the eukaryotes, we found that we had many different copepods, diatom, and green algae. So showing that those dense structures around the microplastic are forming independent ecological niches. So we have a microenvironment in the larger environment, the larger ecosystem when the microplastic are. As I said, we were interested to see if among those microorganisms, we had some pathogens. Starting with the potential pathogenic bacteria, we identified several bacterial genera that you can see here on this list. Some names might ring a bell. So we found some Salmonella species, some Vibrios, but we were very surprised to see also potential pathogenic eukaryotes. We found this species of copepods that you can see here, the Caligus rosacreci, which is a very well-known parasite of fish, and it causes great damage in aquaculture farm. So here again, we can wonder if this potential pathogen might travel in different ecosystems and contaminate and be released in different ecosystems, and what are the consequences on fish populations. And also we identified this genera of fungi that you can see on the left, which has been demonstrated as a very dangerous parasite for amphibian around the world. So it is quite a concern to identify those potential pathogens in microplastic that can travel from different ecosystems. But the biggest question is, okay, we found potential pathogens, but are they really pathogenic? And this is a question scientific which are investigated. Microbial community attached to plastics should better understand and dig into these questions, because many studies demonstrated and raised the alarm that there might be potential pathogens on the biofilm, but we still don't really know if those species might have the capacity to really infect fish or human. So to try to have more information about that, we investigated the diversity of virulence genes and resistant genes in the biofilm foreign samples. So the graph might be a bit scary, but don't worry, it's quite simple. In fact, you have on the left the list of all the resistance and virulence genes that we identified. And on the left panel, it is when we found them in the biofilm. And on the right panel, it's when they are present in water. And the darker the color, the more abundant those genes are in the samples. So the first thing that we see is clearly that in the biofilm on the left, we have more dark colors. So we have an enrichment of resistance and virulence genes in the biofilm. So this was quite also expected because we had greater abundance of bacteria, as you recall from the beginning. But when we dig into the diversity of those genes, we found that we identified several families of genes conferring resistance to many antibiotics that are commonly used in medicine and in agriculture. So this is, again, worrying because those genes might spread in the environment and therefore have consequence on our resistance to those treatments. And we know it is a very an important issue in medicine today. And when we look at the diversity of virulence genes, we were quite surprised because we found many potential pathogens in the biofilm, but we didn't really find virulence genes. So we were not able to say, okay, we have pathogens and they have a lot of virulence genes. We cannot say that. Most of the virulences that we found were linked to some activities which are not so much considered as pathogenic in this case, because they are linked to the adhesion of the bacteria on the biofilm. So the real question is, in the biofilm, are the pathogen really pathogenic, or do they sometimes like lose their pathogenicity? Are they really capable of infecting fish or human? And this is a question we really need to better understand today, to better understand if those pathogens can spread in the ecosystems. So to sum up a bit, what we saw today, we saw that the colonization of microplastic is not only done by bacteria, but also by different kind of microorganisms, among which we can have some potential pathogens and some resistance and virulence genes. And this poses the risk of the dissemination of both those pathogens and also of those genes of interest. But also, many questions remain about the impact of this colonization of microorganisms on the microplastic. For example, what impact does it have on the degradation? We saw those very dense structure around the plastic particles, so maybe it has an impact on the capacity of UV light to degrade those plastic because they are more like sheltered in those communities. It might have also an impact on the efficiency of the recruitment of bacteria which are capable of degrading those plastics. Same, we saw that we have those very dense community and very diverse, but we still don't know what are the interactions between the microorganisms in those communities. Maybe there's some competition, maybe there are some exchange, some predation, and how those might affect the growth, the development, the photosynthesis activity of the phytoplankton and zooplankton species that we found attached to the microplastic. And finally, some researchers are having some concern about the impact of so much communities at the plastisphere on the biochemical cycle, because those species might have the capacity to recycle nutrient and recycle carbon, but how much do they recycle and how much is back into the ecosystem is still something that we have to better understand, to put in models to better understand the evolution of aquatic ecosystem, which are threatened by microplastic, but also global change, ocean acidification, and many other things. So to give small take-home message, I would like you to think that the fact that marine animals are eating plastic litter is only the emerged part of the iceberg. Microplastic and their attached blowfish might be a great threat to aquatic wildlife, human health, and all marine ecosystem equilibrium. And we need to better estimate what are the impacts of this plastisphere on the ecosystem and seek for better solutions to prevent the accumulation of those plastic in aquatic ecosystem. And with that, I thank you very much for your attention and be very happy to take any of your questions.
Thank you very much, Elsa. You have given us a lot of valuable information to consider, and I believe these will spark further in discussions and research in this area. This one is coming. What types of microorganisms are most commonly found colonizing microplastics in aquatic environments? And what are the implications for the aquatic ecosystems?
So this is a very interesting question because to date, most studies investigated bacteria species which are attached to the microplastic. So for now, we can say that bacteria are the most abundant, but we don't really know because some other species are simply not investigated at all. We have very, very scarce information on the viral composition, the viral components of the plastisphere. And so, yes, to date, bacteria are the most abundant, but it might also be something else if we have the tools to look for who is actually here and how they can impact human health. The main risk might come from the transportation of organism from one ecosystem to the other, because those microorganisms might not stay forever attached to the biofilm and might be just spread in the environment, and that way we might enter in contact with us, human, but they can also potentially get through us through the food chain as well. But this is still has to be investigated more deeply to bring final answers to that.
Thank you, Elsa. You read that we have another questions. We might take it this one. Do you have any idea of bacterial spores and microplastic?
Actually, not really. We investigated only the diversity of it, but it is certain that we have essential function going on in the plastisphere, and that the bacteria are replicating, if that is the sense of the question.
Hope so. Thank you, Elsa. Thank you for your presentation.
Thank you.
And it's time now to give the floor to our next presenter. So I have the pleasure to present Stephanie Bedhomme. She is a CNRS researcher at the Center for Functional and Evolutionary Ecology in Montpellier. During her career, she has been awarded an ERC Consolidator grant. Stephanie is an evolutionary biologist, and her main interest is in the evolution of bacterial genomes, with a particular focus on horizontal gene transfer and antibiotic resistance gene circulations. Stephanie's presentation today is targeting a critical question. Are microplastics shuttles for pathogenic bacteria in marine environments? Please, Stephanie, the floor is yours.
Thank you, Lara, for the nice introduction. Thank you to everybody for being here to this Green Week event today. So my presentation is going to be a bit complementary and in the continuation of what Elsa just told us. And I wanted to start a bit further away or by talking about the origin of these microplastics. So it's evaluated that more than 400 million of tons of plastics are produced each year in the world, and more than 50 million tons in the European Union. And from these plastics that are produced, they have a very large diversity of use, but they can be degraded, and the degradation products are either microplastics, which are plastic fragments from 1 micrometer to 5 millimeter, or nanoplastics that are fragments below 1 micrometers. And it's evaluated that each year 12 million tons of microplastics are produced and reach the ocean. So another notion that probably we have to have in mind is that this pollution by microplastics is not homogeneous. So if we now look at the concentration of microplastics that is evaluated, and the slide, the graph we are seeing is a map where predictions have been made through modeling and knowing the source of the microplastics and where the contamination is higher. And so it's first estimated that globally there is between 15 and 50 trillion plastic particles floating on the surface of the world ocean. But the other important information on this slide is that there are hotspots for microplastics contamination. And you can see that the Mediterranean Sea is one of them, but also the Yellow Sea, so between Korea and China. And there are, of course, also variation in the open oceans. So if we now think about the effects of microplastics on human health, we already had some examples in previous talks, but it's important to think that the oceanic microplastics can enter the human body either via the food chain and the digestive tract, or via ocean spray and the respiratory tracts. And there are going to be direct effects of these microplastics by chemical and physical interactions between the particles and the cells they are in contact with. So for example, the gut or the lung cells. Regarding the the entry in the human body via the food chain, there has been many estimates of the number of microparticles that are contained, for example, in seafood, such as mussels or oysters and clams. So Tanja has shown that, and here on the map that I previously used, you have now indicated different places where these estimates have been done. They vary also widely, and they kind of correlate with the intensity of the contamination in the different places. But now I would like to go to potential indirect effects. And these indirect effects are going to be through all the elements, all the microorganisms, all the chemicals that can fix or adhere or absorb on the microplastics. Indeed, when a microplastic is going to reach a natural environment, it's not going to stay naked or just as a microplastic particle. There is going to be a formation of a hard corona, which is made of organic material, of proteins, of other molecules, such as, for example, so biomolecules, but also metal ions. Then there is going to be a second layer, which is the soft corona, where there are going to be a more exchange with the environment. And once this highly nutritious corona is formed, then bacteria are starting to be attracted and to colonize the microplastics. And so there is going to be a succession of colonizers. So some species of bacteria are going to colonize first, some are more specialized in colonizing already mature biofilm. But in the end, we are going to have a fully mature biofilm. And so this, the organic material plus the microplastic is called, plus the bacteria, sorry, is called the plastisphere. And if we now zoom in this plastisphere, you can see that some of the bacteria that are there are known to be pathogens, either pathogens of fishes or of seafood in general, or human pathogens. And so the question is raised of whether this is a risk or a danger for human health directly, or for the food provisioning of humans. So there are now quite a number of studies that have analyzed the composition of the biofilms on the microplastics, and many of these studies have found species such as Vibrio and have raised the question of the potential of the microplastic of being vectors of human and seafood pathogens. However, answering this question requires to investigate more precise and specific questions. The first of them is that we need to understand the factors that are influencing the attachment rate of the microplastics. For example, factors such as the intraspecific variability. So within a bacteria species, are all the strains going to colonize equally? Does the polymer of the microplastic influence this attachment rate, or the stage of colonization of the biofilm on the microplastics? But also we need to know how stable the attachment is, which is another way of formulating the question, how far away can a microplastic transport a pathogenic bacteria? So is it going to be from very close, spatially very close ecosystems, or can we imagine very long transportation, very long distance transportation? And another important question is, do the pathogenic bacteria fixed on microplastic constitute a better infection starter than seawater bacteria or bacteria that are fixed to other floating particles? So the results I'm going to present today are mainly on the first question. And to answer this question, we have chosen as biological model Vibrio angularum, which is a fish pathogen that is damaging economies of fisheries and aquaculture. We have gathered a collection of 16 strains covering a diversity of places, host, and time of isolation, as you can see on the map on the right. And we have labeled these 16 strains with a fluorescent protein, such that we can see the bacteria attaching to the microplastic, as, for example, on this image where they're attaching to a polystyrene sphere. As microplastic models, we have used polypropylene and polyethylene terephthalate particles that have been produced by milling of larger pieces of plastics, and also polystyrene beads that are manufactured and round regular shape. We have incubated microplastics and fluorescently labeled Vibrio anguillarum for 24 hours at 30 degrees. And we have used fluorescence microscopy to evaluate the attachment rate. So we have combined the information of bright field and green fluorescence filter to quantify the proportion of the bacteria that have attached to the microplastics. And so here are the results for the 3 different types of particle we have used. So you can see that PET particles have a higher percentage of bacteria attached to them than PP particles, and PS particles have way lower attachment rate. So we have here a strong effect of the polymer composing the microplastics. But also you can see that within each polymer, we have a huge variation. And this variation is probably due to several factors, but one of them is actually the strain of Vibrio anguillarum that has been used for the experiment. So here now on the right of the slide, you have the same data as on the left, but they are this time separated by Vibrio strain. And you can see that each Vibrio strain has a different rate of attachment and that there is a quite large variation within the species for the attachment rate. We can also see that it's the same strains that are good attachers on PET and on PP. So it seems to be a specificity of the strain, and that the polymer does not matter that much for that. So to sum up on this part, we have an attachment rate that varies strongly with the polymer. I would like to attract your attention here on the fact that it might also vary with other physical properties of the microplastics, because if you remember, these 3 microplastics haven't been obtained in the same way. So PET and PP are milled particles, so they are very, they have a high rugosity, a lot of unfructuosity, where potentially bacteria can fix and multiply, whereas the polystyrenes are round shape, and that might explain the difference between these 2 categories. So this would need to be investigated further. But in the end, it means that the vector potential of microplastics depends on the polymer constituting the microplastics. We have also seen that the attachment rate varies strongly for different strains of the same species. And so it means that it's going to be difficult to establish the attachment rate of a species. And then, so that first experiment was on naked particles, so particles that we had just obtained and that we incubated. But we were also interested in knowing what was happening on particles that were previously colonized in the film, in the field. And so for that, we incubated particles in the same way that Elsa presented in 3 different places along the western Mediterranean coast of France. So we had incubations in Carvenon Harbor, which is a leisure harbor close to Montpellier. We had incubation in Bassin de Thau, which is a zone of oyster production. It's a laguna. And then we incubated particles close to Bagnols-sur-Mer in a marine reserve, which is supposed to be a protected environment. We incubated the 3 same types of particles as before, and we added non-plastic controls, which were glass beads. So these incubations were for a month. And in parallel, we also analyzed the composition of the biofilm in the same way as Elsa presented. And we know that the taxonomic composition of the microplastics associated to the biofilm differ with the location. We brought back these particles incubated in the field, and we also had sterile controls, but then we did exactly the same experiment as before, which is an incubation with fluorescently marked Vibrio anguillarum. And this time we only used 3 strains of Vibrio anguillarum. And the results I'm going to present are in the form of the difference of the percentage of bacteria attached to microplastics between field colonized and their control. And so here are the results. So all the dots that are above the lines means that there is more attachment on field colonized microplastic, whereas the dots that are below the line are, indicate more attachment on the control microplastics. So the information that is very clear here is that the result strongly depends on the polymer. So we have less attachment on PET and PP for field colonized particles and more attachment for PS and glass beads. So the presence of a mature biofilm on the microplastic is changing the attachment of Vibrio anguillarum. The direction of the change depends on the substrate, and it results in a more homogeneous attachment rate. It seems to erase the differences, and it's kind of logic. So this biofilm that has formed is kind of erasing the differences of physicochemical properties of the particle. It might also be due to chemical interactions between the bacteria in the biofilms and the Vibrio anguillarum that we test. And the effect is quite similar for the 3 locations and the 3 strains of Vibrio anguillarum. So it seems to be a general effect of the presence of the biofilm that we are detecting here. So the take-home messages are that the fish pathogen Vibrio anguillarum attaches to microplastics, either naked or with a mature biofilm already formed on them. The attachment rate varies with the polymer and potentially with other physical properties of the particle, with Vibrio anguillarum strains, and with the presence of a mature biofilm on the particle. So for broader conclusion and future direction, the microplastics definitely have the potential to be vectors of pathogenic bacteria, but quantifying the risk it represents will be difficult because we have seen that there are many factors that can influence this attachment rate, and also requires to investigate further questions. So some I mentioned earlier, so the stability of the attachment, how far away can a microplastic transport a pathogenic bacteria? Also, I think it's very important to really compare the risk that is presented by the pathogenic bacteria that are fixed to the microplastic to the risk presented by these pathogenic bacteria that are in the seawater or that are fixed on other floating particles that are more natural, like wood, sand, or shell fragment. And to see the proportion of particles floating that are microplastics, how the community on them change, because it's not because there are pathogenic bacteria in the microplastic that they represent a very big risk. We have to be careful not to be alarmist, but we have also to really evaluate the risk that they represent. And so finally, I would like to thank you for your attention and many people who made this study possible.
Thank you. Thank you very much, Stephanie. Now, I guess we have some questions from the public for Stephanie. What are the most critical factors that determine whether microplastics will act as effective carriers of pathogenic bacteria?
Well, I think the first factor is how much pathogenic bacteria fix or adhere to the plastic. The second critical factor is how stable this interaction is, and so how long they stay together, which is going to influence how far away this particle can be transported and whether it's going to actually reach another environment or be eaten by a filterer, for example. So to me, that's really the two particular. So something that Elsa mentioned also is how much the fact that this pathogenic bacteria and other bacteria are fixing is going to change the behavior of the particle in terms of floating or sinking and be transported. So that's probably the main factors.
Thank you. We have another question. How might the findings on microplastics as vectors for pathogens influence our approach to managing marine pollution and the public health?
Well, that's quite a difficult question.
We hope so.
Yeah, in terms of marine pollution, I mean, I think we have seen today, and there are many studies around that, it's pretty clear that plastics in general and microplastics present dangers, whether it is directly or whether it is through the potential pathogen that they carry. So to me, all these findings should go into increasing the pressure to reduce this production by microplastics. And I think like the starting point to reduce this production would be ideally the reduction of the production of plastics themselves, because the microplastics are mainly a degradation of plastics and not that much. So, I mean, the particles that are initially produced as microparticles are also important. It's important to reduce their production and their use, but it's the degradation product are the big bulk of the problem. So I don't see how we can reduce this marine production without reducing the initial production of plastics. So there are some efforts clearly that are made in this direction, but we should really go further on that. And in terms of managing public health, I think we should be careful for now, and I think it's a bit too early. We don't have really assessment of the risk, the pathogenic bacteria that are carried by microplastic represent to public health. We have evidence that there are antibiotic resistance genes, for example, that are on these microplastics. We know that biofilms are a hotspot for horizontal gene transfer, that there might be a place where the genes are transferred. So for that, it's already quite clear that they represent an additional risk for antibiotic resistance gene propagation. How to manage that, it's the same. I think we should find a way to reduce their presence.
Thank you, Stephanie. So it's time now to present our two experts that have been preparing the next presentation. I have the pleasure to present Professor Tatjana Parac-Vogt. He is a full professor of chemistry at KU Leuven and the head of the bioinorganic chemistry group. She has a multidisciplinary expertise in the fields of bioinorganic chemistry, coordination chemistry, nanochemistry, and spectroscopy. And then Ali Moussawi, he is a postdoctoral researcher at the Department of Chemistry at KU Leuven. After completing his PhD research at Lavoisier Institute of Versailles, in France, he is now investigating the interaction of metals and metal complexes with the biological systems. So now I'll give the floor to Ali. Thank you.
Thank you so much for the introduction, and I'd like to thank all of you for joining us today as we discuss an increasingly alarming issue that affects not just our environment, but our health and the future of our planet. The contamination of our world by microplastics, and more specifically, the grave risk posed by heavy metals and metal ions that adsorb into these microplastics, that adsorb into these microplastics. While microplastics alone are a growing concern, the combination with toxic heavy metals creates a cocktail of dangers that we can no longer afford to ignore. Metals are essential for the human body, playing critical roles in various biological processes. For example, iron is vital for the transportation of oxygen in the blood, magnesium supports muscles and nerve functions, and zinc is crucial for immune system and the health. However, exposure to heavy metals like lead, mercury, cadmium, and others pose significant health risks. And like the essential metals, these heavy metals accumulate in the body, leading to toxicity. Chronic exposure can damage vital organs, including the brain, the kidneys, the liver, and so on. And it can result in neurological disorders, cardiovascular diseases, and more. Thus, protecting against these heavy metal exposures is crucial to preserving the human health. As explained by my colleagues, microplastics are tiny plastic particles, typically less than 5 millimeters in size or in diameter. They can be made from several materials and thus have the affinity to absorb numerous toxins and behave as carriers for bacteria and allergens. What makes this issue even more concerning is that microplastics have a high surface area to their volume, and thus and their rough surfaces, allowing them to absorb heavy metals and other pollutants from the surrounding. This combination creates a perfect storm of environmental and human health risks. When microplastics absorb heavy metals, they become highly toxic to marine life. For instance, microplastics carrying adsorbed metals can be ingested by oysters, leading to reduced growth and reproductive rates. Over time, these impacts can cause population decline, disruption of their entire ecosystems, and the services that they provide, such as the food source and natural water filtration. The impact of microplastics in aquatic organisms affect their biological ingestion process, causing clogging and indigestion, and in adverse conditions, they hinder the biological processes, resulting in toxicity, immune dysfunction, reproductive abnormalities, and so on. And in final cases, death. In addition to this, the absorption of microplastics result in bioaccumulation and biomagnification, which is very concerning. This biomagnification across the food chain makes its route to the human body. When we eat seafood or crops contaminated with microplastics that have heavy metals adsorbed to them, we are unconsciously introducing these toxic substances into our bodies, leading to severe issues which can persist for decades, threatening our health and the future. Now that we understand the significant impact and the risk posed by the combination of microplastics and the metal ions, I will present some of our recent findings on the adsorption of metals into various microplastics collected from 3 different locations in the Mediterranean. Samples from these locations were tested by our colleague Stephanie in the previous slide, in the previous presentation, and her team, who showed that these plastics are carriers for pathogenic bacteria and so on. This combined threat from the different pathogens like bacteria, allergens, pathogens, and heavy metals is highly concerning, and we should keep a focus and then concentrate on finding a solution. The samples we recovered from our collaborators from Stephanie in France, and the microplastics are isolated by filtration in the first stage. And then here we visualize their shape and size by scanning electron microscopy. Polyethylene and polyethylene terephthalate and polypropylene have different properties owing to their unique composition. And as can be seen, they display different surfaces and morphologies. Polyethylene terephthalate, PET, is usually used for food packaging, textile, fabrics, and electronic components, while polypropylene is used for plastic bottles, food containers, and automotive parts. So the isolated particles were also visualized by transition electron microscopy in combination with energy dispersive X-ray spectroscopy. These allow us to visualize or determine the nature and dispersion of metals or metal ions on the surface of these microplastics. As can be seen here, on the left, we see the scanning electron microscopy image of the particles, and here there's the structure of the polyethylene terephthalate. The gray image shows the actual particle, and then the red is the map of the carbon that forms this. If we look for other compositions on this, or other metals or elements on this particle, we see that there are copper, iron, and other metals. I visualized here copper and iron because they are the most spread in that particular sample. If we look into other particles like polypropylene, we see as well the carbon that makes the backbone or the chain or that makes this polymer, the polypropylene. But we also see other elements like aluminum, iron, magnesium. Magnesium in this case has the same exact pattern that we see for oxygen, and this suggests that we have magnesium oxide. A small research about this sample that was isolated from the lagoon where the oyster farm is, we learned that oyster farmers, they introduce magnesium oxide into the environment there that helps in hardening the shells of oysters, and it is an essential component in their growth. So now we know that these microplastics, they don't absorb anything. They absorb everything and anything, from pathogens to microplastics, even the nutrients that we add for these oysters. Now, if we want to check the dependent metal absorption on these microplastics, we checked different plastics. We checked polypropylene, polystyrene, polyethylene terephthalate, and we put a control of the glass beads. We see that different microplastics have different affinities to absorb metal ions, with polypropylene having high affinity due probably to their rough surfaces and thus the high surface area to their volume. Additionally, the absorption is quite seasonal dependent. So for instance, if you check these graphs, they show that there is high absorption during summer in these regions. And then with the highest absorption on plastics is close to the harbor, Carnot's harbor, which is roughly at a site or a location that has a lot of boats, fishing boats, contamination from all the buildings surrounding it. And we see there's a lot of contamination from metal ions in that region. If we test the same regions, for instance here, the marine reserve, Bagnols-sur-Mer, where we checked the absorption of different metal ions during summer, spring, and winter, we see that there is a higher absorption of these metals during the summer incubation. This can be due to several conditions. For instance, the degradation of these microplastics under UV radiation from the sun, or the high temperature, which enhances the mobility of both the microplastics and the metal ions in the water, increasing the chance of them clashing and then absorbing. Increased biological activity, as Stephanie mentioned, they transfer pathogens and they transfer metal ions and other allergens. So warmer temperature stimulates biological activity in the water bodies, which may enhance the absorption capacity of microplastics for metal ions. Changes in the microplastics, again, from the temperature or the UV radiation, can cause physical changes in microplastics, which will increase their roughness and surface area, making them more prone for absorption. So in the end, we have to focus on the major issue here. Microplastics contaminated with metal ions is a huge concern at the moment. And in combination to everything that we saw from my colleagues, this has to be addressed and addressed ASAP. So why should we address this issue? Because metal contaminated microplastics in our water are spreading everywhere, in freshwater bodies, in saltwater bodies, in, for instance, oyster farms that we consume on a regular basis, and so on. And they cause degradation of our environment. The degradation of these microplastics that have metal ions that have been transported from one location to another leads to the release of these metal ions in different locations, destroying the environment in several locations. And this can cause a health burden on the society. The increased medical bills, the human health at risk, especially for kids that go into these water bodies in summer or like at any point. So a lot of actions and solutions need to be put into action as soon as possible. For instance, reducing the plastic use. We have been saying for decades that we need to reduce plastics because of their high demand and high consumption. We need to find alternatives. We need to reuse these. So improve waste management, try to recycle, and so on. We have to research and monitor all these effects caused by the microplastics that are combined with the metal ions and so on. And we need to increase the public awareness. These microplastics are a huge concern at the moment, and we can't leave it. And international cooperation to fight this is needed. So through Imptox, we are trying with teams in Belgium, in Serbia, in France, in other countries, in Austria. We're trying to combat this to the best of our efforts. So in conclusion, the issue of heavy metals absorbed into microplastics is a silent but a serious threat to our health and our environment and our future. The stakes are very high and the time to act is now. So by taking steps into reducing these microplastics and their use and their release into the environment And by raising public awareness, we can begin to mitigate the dangers posed by these toxic pollutants. And failure to do so will not only endanger our own health, but will also jeopardize the health of the future generations and the very ecosystems that sustain life on Earth. So finally, I'd like to thank you for your attention, and I hope that this talk has highlighted the importance of addressing this critical issue before it's too late. I'm happy to take your questions.
Could you elaborate on the specific conditions under which microplastics most effectively absorb and transport metal ions in aquatic environments?
Well, that's an interesting question, but that we can answer. So to elaborate on that, like there's a lot of conditions. For instance, I mentioned that these microplastics have high surface area and size, like they have high surface area to size ratio or to volume. And the smaller microplastics generally have larger, like these larger surfaces that absorb more metal ions. And then they have different chemical compositions, as I presented the structure of polypropylene and polyethylene terephthalate. These have different functional groups. For example, the polar functional groups can form bonds or electrostatic attraction with these metal ions and then increase the affinity of these microplastics to absorb metal ions. Presence of organic matter, for instance, the pathogens or the biofilms that someone asked about, they can form around these or like deposit around these microplastics, and then these can also help in absorbing metal ions or not. They can be competing with them or helping them in binding to the microplastics. And then the higher the metal ion concentration in a water body, for instance, as we compared the harbor where there's a lot of contamination in comparison to a marine reserve, the higher the metal ion concentration, the higher where it will be absorbed onto the microplastics. I hope that
temperature as well. Temperature.
Yeah. So higher temperature will increase the collision and then the binding.
Thank you, Ali. We have another questions from the public worried about the fact that we might drink water with heavy metals, tap water or bottled water.
Well, usually these water, like water, if you get it from supermarkets or like you buy these, they usually are tested and then you have like the metal composition in them. So you should not be very concerned. But if you're in the environment at a river, in a pond, and then you consume water, I would be concerned because these areas, they have a lot of contamination, not only just metal ions, but other things. But metal ions specifically because even in very low concentrations, they cause severe damages. And in higher concentrations, you need like hospital visits using chelating agents to remove these metal ions from your body. But if you're concerned about water reaching your home, I would not be that concerned at the moment.
Okay, thank you very much.
You're welcome.
So now it is time to move on, and I have the pleasure to present you our next speaker, Professor Asselman. She is the head of the Bloom Growth Research Lab at Ghent University. She coordinates the multidisciplinary research facilities located at Ostend Science Park. Professor Asselman is a marine biotechnology expert focused on the biodiscovery, microplastics, and sustainable aquaculture. And her interest focuses on human and environmental health effects of chemical pollution in water, including micro and nanoplastics. Jana's presentation today is micro and nanoplastic pollution in the environment and links between environmental health and human health. Please, Jana, the floor is yours.
Thank you, Laura, for the introduction. Let me just share my screen. There we go. So I'm the last speaker of today, and I would like to take the time to put all of the things that we've seen so far a bit more into perspective and how it links from environmental risks to human health risks. Yeah? So what we've seen today already is when we talk about plastics in the environment, for human health, we're mostly worried about the potential direct effects for the water when we use it as drinking water, for food, particularly thinking about seafood, but also for the blue spaces, rivers and oceans that we use as sources of recreation and tourism. But of course, as already addressed today, there's also much more. The ocean provides numerous ecosystem services, and we also see that plastics also impact those services that we use for health. Think about primary production, oxygen generation, biodiversity, and biodiscovery. So what does all of that mean if we try to put all of these numbers together? Well, let's start with looking at plastics in the water we use in our rivers and oceans. And I'm going to share with you some highlights of our published work. So here we focused on characterizing microplastic pollution in the urbanized region of Flanders, which is a very good example of a local scale microplastic pollution because we have such high levels of urbanization. We used infrared spectroscopy to get information on the plastic concentration. Of course, because of the analytical limits, we're limited to particles larger than 20-25 micrometers. We also were able to identify polymers as well as length and width of each particle. And as you can see here, we analyzed the microplastics in the rivers all across Flanders, and we analyzed them both in wet season and in dry season. Why? Because we know that rain, but also storm overflows can significantly impact microplastic concentration in the surface water. And what you can see here is that it varies all across Belgium, but actually that the average levels are not that high. What do I mean with that? Basically, if we look at surface water microplastic pollution, we look at levels all across Flanders from about 0.36 microplastic particles per liter, so less than 1 particle. And I'll come back later to what all of these numbers mean with regards to human health. If we look at the water that we then use for drinking water, it was already addressed as well during the questions. So in Belgium, we use 3 different sources of drinking water. We use groundwater, we use surface water, and we also use purified wastewater. As expected, of course, the purified wastewater contains the largest amount of microplastic, and then followed by surface water and the lowest amount in groundwater. But again, these numbers are not extremely high. Looking here into these water categories, into polymer types, we saw quite some differences depending on the location of the tap water. And we looked into tap water in particular. For example, in Ghent, we found quite a diversity of polymers present in the tap water, whereas in Kortrijk, we saw that the tap water was dominated by PET and PP. With regards to sizes of the plastic particles that we found in the tap water, the majority of the plastic particles was actually located in terms of the size classes between 25 micrometers to 100 micrometers. Please be aware that we can't detect particles at this point smaller than 25 micrometers in numbers because of analytical constraints. So basically, when we look at plastics in drinking water, we're already going 10 times lower than in surface water. We're adding 0.02 to 0.05 microplastic particles per liter in our drinking water on average. Then one of the last direct effects we can get from microplastics in the environment, particularly in blue spaces, is of course food. We use a lot of seafood, we eat a lot of seafood. And these are some results for mussels, oysters. We also added a reference, so a marine invertebrate. And as you can see, these particles numbers vary across these different species. If we put this more into context about seafood, we're of course talking a little bit more into kilograms. So if we're talking about shrimps, for example, white mussels, commercial mussels, etc., we see these numbers varying. And what we often see is that the variation is often less in commercial forms than in wild mussels, because this is often because of the way they're cultured or the locations where they're cultured, where we see that wild mussels inhabit both extremely or more polluted ecosystems versus also more unpolluted ecosystems in the marine environment. We also see, of course, that if we compare pre and post depuration, the number of particles per gram of tissue decreases, which is not unexpected, as most of the plastic accumulate in digestive or digestive-like tracts in these types of animals. So basically, if we're talking about food, we're also, again, in this same ballpark number. So what does this mean? What do these numbers mean for human health? Well, basically, with this work, we're seeing that aquatic environments and sources, while they do contain plastic, if we look at our health, they're likely not the major or the largest contributor of direct exposure routes. If we're looking at addressing human health risks and the amount of plastics that you and I are exposed to on a day-to-day basis, we need to really look closer to home. We need to look at food, drinks, particularly packaging as well, clothes, fabrics, air. Now, why do I say this? Because we're also looked at the amount of plastic in our domestic wastewater. This is a proxy a little bit to what we're exposed to on a daily basis, because our domestic wastewater is a collection of all the water from our dishwasher, washer, shower, toilet, sink. So it picks up all the pollution that's present in our environment or that we ingested at some point. And it's even actually an understatement, because some of the pollution in our house will eventually also not end up in the wastewater. And what we saw in the wastewater is that we found values between 1 to 40 microplastic particles per liter. So remember, from direct exposure in the water, the highest value we got was 1. So we're going here from 1 to 40. So we're 40 times higher in our house than the plastics in the environment. We quantified this basically on the number of plastic particles a person generates in terms of wastewater. So that was anywhere between 350 to more than 1,600 particles per day per person. So that's a lot of particles we're generating. So looking at links between environment and human health, we as humans have a significant impact on that aquatic environment. But luckily for us, the direct exposure that we get from that same environment is rather limited. Now, it means we need to look closer at home. And I want to highlight just very briefly why it's important for us to address this closer at home route. So one of the things we've been doing together with the colleagues here present today is we looked at microplastic particles and what they do in different types of human cell lines: lung cells, gut cells, liver cells. We cultured them in different scenarios. We looked at biological responses. Professor Rajkovic already mentioned some of those, but also at internalization and localization. And that's something that I wanted to share with you today. So looking at these very different graphs, let's focus on the top ones first. What you see here is increasing number of particles that human cell lines were exposed to. And for all of these lines, each line is a different cell. We actually see that, which is rather normal, as you increase concentration, we see more particles taken up by our cells. So our cells clearly take up particles. This was what we expected. It's confirmed by tons of other studies as well. But what's more striking is what I like to share with you here. So after we did all of these uptake experiments, we actually started culturing the cells for another 2 weeks without exposure to microplastics. And we actually saw, you can see that very clearly here, that even after 2 weeks, the plastics don't get out that easily. Whatever we take up in our cells stays in there, and it takes quite a long time for us to get it out of our cells. So that's really important because it raises significant questions for human health as to how many plastic particles can our cells actually cope with, knowing that we're exposed via so many different routes to plastic particles on a daily basis. So with all of that in mind, of course, raises the question, plastics in the environment, it's clear that they're not the major issue for the health of you and I. So why should we care then? Why should we care about plastics in the environment? Well, first of all, they have significant interactions with all of other pollutants. We've already seen by the previous talks that they might be a hotspot for antibiotic resistant genes. They might interact with harmful algal blooms, and they significantly affect our marine and aquatic ecosystems in terms of biodiversity. And that biodiversity is super important. People always find that very abstract. Why is biodiversity important? What's the benefit of biodiversity to me as an individual person? Well, let me show you that. The biodiversity for you as an individual person is basically this. Over the past decade, we've got numerous new medicines from the marine environment that treat one of the most challenging diseases that we face as a society. A lot of new cancer drugs are being developed on a yearly basis from the biodiversity in the ocean. I'd like to say that most of the innovative medicines, we don't invent them from scratch. They don't come from our brain. They come from the marine environment, and we use that environment, that biodiversity, as an inspiration to treat several important diseases. It's an important source of curative healthcare. But at the same time, what we've also seen is that people who live close to blue spaces, and particularly marine environments, they live longer because those marine environments contain lots of bioactive molecules that basically function as some sort of preventive healthcare. And we risk losing all of that because plastic environments threaten marine biodiversity. So basically, while there are very small risks of plastics in the environment directly to you and I, mitigating aquatic plastic pollution will be crucial for our health as a society and as a species because they provide so much in terms of ecosystem services, in terms of new medicines, in terms of preventive and curative healthcare. Thank you for your attention. And I'll be happy to take any questions now.
Thank you, Jana, for your presentation. We will have a session of questions that might be for all of our speakers. So we have one here for the University of Ghent. Can you comment on this first statement about the influence of microplastic infectivity of pathogens?
Yeah, well, what we have seen also in our own studies is that number of key foodborne pathogens, including Campylobacter and Listeria, have been actually exercising higher infectivity capacity towards the human cells in vitro studies, as well as the gene expression of the virulence genes, particularly when they were in contact with microplastics. So yes, there is a certain increase. There is a change, which is negative. Basically, if one would like to summarize this very shortly, it is a potential that pathogens become more dangerous after exposure to certain stressors. And in this case, stressor might be a microplastic particle. But this could also be because of the closer cell-to-cell contact of bacteria when they are forming communities on the top of the microplastic surface.
Thank you, Andrea. I have here a question for Jana, the last presenter. The question is, what research gaps exist in understanding the full impact of micro and nanoplastic pollution on human health? And what future studies do you think are more critical?
I think what's extremely critical at this point are two things, is exposure scenarios. We know there's a lot of data on acute effects, so responses in 24 or 48 hours. But for our health, what's happening is that we're exposed on a daily basis to rather low doses of microplastics. So what we urgently need is lots and lots of data on long-term chronic exposure scenarios, and how this would affect our health if we keep on accumulating low effects or low doses of microplastics on a daily basis. And then the second part, and it follows up on that, is how does this microplastic burden adds relatively to all of the other burdens that we're experiencing as humans with regards to chemical pollution, dust pollution, etc. How big or how large is this contribution? I think those are the two important research gaps that we need to address in the next couple of years.
Thank you, Jana. I would like to give the floor to our coordinator, Imptox coordinator, Tanja, that was kicked out of the Zoom platform before. And I'm sure that she wanted to say something. In particular, I would like to ask, in your research, in the Imptox research, what challenges we encountered in accurately quantifying micro and nanoplastics in diverse water environments, and how have you addressed them?
Hi, thank you, Laura. Apologies to everyone for technical issues with the Zoom platform for me. Today, I'm sorry I could not conclude on the topic, but most of the speakers addressed a lot of things that we actually have seen and confirmed in our study, particularly related to the contamination of seafood with microplastics, as you have mentioned, and I have also tried to cover it a bit. We are experiencing a lot of technical issues. For now, we know that the most commonly used methods have its limitations. One of the limitations is the size limit, but we also have to say that there are no appropriate standards. The methodology should be improved and more standardized, and more reference materials should be used. We are still seeing a lot of inter-laboratory variation when characterizing and quantifying microplastics. And when it comes to the nanoplastics, we are far, far, far from being able to fully characterize and report its occurrence in the numbers, in like size ranges, etc. So when it comes to our own research, I would say that we have made this nice progress regarding the sample preparation. We have optimized our digestion protocols. I believe also within newly available reference materials for microplastics, also improved our FTIR quantification measurements. In this regard, we are a step towards being where we should be when it comes to the microplastics analysis in complex matrices such as food and biological materials. But when it comes to the nanoplastics, I have to say that we still have a lot of things to do, and we are not yet there yet. So I really want to thank to all the participants and for very nice overview of our research efforts in Imptox. As I think Stephanie said nicely, we do not want to raise unnecessary alarmism, but we should actually report our data that indeed show that we should think of the hazard and of the risks associated with the presence of these particles widely in the environment.
Thank you very much, Tanja. As we draw this remarkable event to close, I would like to thank all our speakers for their excellent presentations and valuable work. The insights you have provided today highlight the urgent need, as said by Tanja, for a coordinated response to tackle the issue of microplastics. And it is clear that this is not just an environmental issue, but one that affects every aspect of our life. And the connection you have drawn between microplastics, human health, and water resilience call for an immediate attention from everybody, policy makers, industry leaders, and researchers alike. So in behalf of the Imptox project and our presenters, I wanted to thank you all for joining us numerous today on our partner event for the European Green Week. I wish you all a beautiful rest of the day. Keep updated on the progress on Imptox and follow us on our social media. Thank you all again for your participation.